Guys, we're back. So let's start talking more about the cancer, and we're going to discuss a lot about progression of cancer. I know you guys are quite interested in it, and this is something that you'll be dealing with a lot. So we're going to go more in depth into what's happening in progression and how cancer occurs. So this is interesting, guys. We are constantly dying. Actually, we're constantly losing cells. We know this. We slog off cells, cells die. Some of the cells are produced in our body are not normal. So they will be destroyed by, those checkpoints and everything else. So what's a homeostasis of your body? Means that you have to replace the cells that you lose. You have to maintain that balance constantly. So what's going to happen if you have a malfunction of normal cellular regulatory mechanisms? Now, these mechanisms will not be killing bad cells. And these cells will be able, now we mentioned, to grow. They will be able to replicate, and I'm going to tell you in a couple of slides, they will actually replicate better than your normal cells. So when this happens is that individuals will develop cancer. So let me talk to you about certain major players in our body that prevent and cause cancer. So what actually causes cancer is on genetic level are proto-oncogenes. Who are the proto-oncogenes? Proto-oncogenes means they can become oncogenes. They're not. So these are the genes that produce proteins that are involved in important functions of your cell. Normal. They work every day. They work very hard for you to be healthy. But if they get mutated, they become oncogenes. They start producing now compounds. They start producing proteins that will lead to cancer. The second group, of course, is mutations of tumor suppressor genes. We talked about it, P53, all these things, retinoblastoma we mentioned, okay? So all these things can be mutated, they don't work well, and cell can develop cancer. But most common cause of cancer, sadly, is spontaneous mutations of our somatic cells. That means that you can obtain a gene during your life that will lead to cancer, okay? So it doesn't have to be inherited. So there can be mutation of somatic genes, which is the most important cause of cancer and most common cause of cancer. Why? Our cells are dividing, and they're dividing very well. And there can be a chance that certain cell avoids detection. And when it avoids detection, it has mutation, it's going to grow better than the others and lead to cancer. So that is another pin. So these mutations are usually caused by a lot of things, but they're also caused by environmental factors that we're going to talk about. So proto-oncogenes is a good old story of Dr. Jekyll and Mr. Hyde, okay? So you have proto-oncogenes which are good. They help us survive. They're constantly producing these proteins that are necessary for our survival. However, if you mutate those proto-oncogenes, they will start producing, you mutate proto-oncogenes. Genes, they will become oncogenes. So they will start producing bad proteins that will actually lead to malignant transformation. So those are the important factors that can actually cause. Now, we have tons of proto-oncogenes in our body, so be aware when you look at somebody's genetic makeup and say proto-oncogene here, proto-oncogene there. Nowadays your patients might go and do counseling, genetic counseling, and now we can even do a DNA profile. They look at it and say, oh my God, proto-oncogene, I'm going to get, onco means cancer. No, they're normal. But if they become oncogenes, there is an issue. And they are very, very common cause. So what's going to cause mutation of your good old proto-oncogene? You heard this, if not, you will very soon, is mutations. So they can end up with point mutations, one single, base pair can be messed up. They can actually have insertion mutations, the whole segments can be inserted in genes. They can have gene amplification, which means that certain abnormal genes will be amplified that they're normally suppressed. Or they can have chromosomal translocation. This is a chop job, okay? It's a butcher's job. What happens is, boom, you take one half of the chromosome, one portion, snap it off and put it on the other and change it, okay? So this all causes a serious problem. Now, what makes proto-oncogene become oncogenes? Accumulation of mutations, okay? It's not just single mutation. Proto-oncogene is not one little gene there, most of the time. It's actually several genes. So mutations can occur in all of these genes in order for proto-oncogene to become an oncogene and cause cancer. So it's not just one. But when a proto-oncogene becomes oncogene and it's very active, this can lead to malignant transformation of the cell. I'll tell you one thing, I'm going to show you that in the next slide. Oncogene is a bad news, okay? Compared to the others, oncogenes are bad news. Now, tumor suppressor genes, we know this, we mentioned this. What's going to happen when we make mutations of those police officers in checkpoints, okay? Those famous tumor suppressor genes. So we know what tumor suppressor genes do. They curtail unregulated progression through the cell cycle, so they actually prevent cancer. They're sitting there looking at the cells are okay, go for it. Bad cell, stop there, okay? So what's going to happen when you have mutation of these genes? They will stop doing control, okay? They will not be able to halt the cell into the specific portion of cell cycle. Cells will progress, and this will lead to neoplastic changes and cancer. Now, one of the mutations that affects the suppressor gene is a loss of function. So what does that mean is that actually the mutation will lead to complete loss of function. Retinoblastoma, guys, complete loss of function, okay? So loss of tumor suppression gene will lead to cancer. And the protein products of tumor suppressor gene we know normally repress cell growth and division. So what's going to happen when tumor suppressor gene is abnormal? Again, the cell can easily move through the cell cycle. Cell can go through cell transformation and develop malignancy. Now, let's talk about good old guardian of the genome. That's how I call it. Is it cool? That's awesome. That's my idea. Okay, so P53. So if Marvel or anybody goes in it, guys, it's me. Yeah, the guardians of the genome. So P53 is kind of awesome. However, if it is so important, it's a target of all the really serious enemies. You know, the enemies that really want to stop P53. They want to give kryptonite to the Superman. Okay, so what do? So it's most frequently inactivated tumor suppressor gene, and it codes for protein P53. We know that protein P53 mutation is commonly implicated in pathogenesis of number of human cancers, because the loss of P53 protein will lead to genetic instability inside the cell. So how does P53 works? We talked about it. It's kind of overview what we talked. It actually regulates gene expression and controls several genes involved in growth regulation. It's going to facilitate DNA repair. So what does it do? It will see that there is a DNA mutation on a DNA. It's going to activate P21. P21 will stop the whole cycle, give time for repair to occur. We mentioned what's going to happen if the repair is not adequate or cell is really abnormal. It will activate apoptosis of damaged cells, will cause cell suicide. So it will move cells to apoptosis, and cell would die. That's, guys, the most, let's say, most appropriate way for cells to die. When cells die through apoptosis, I'm going to tell you this toward the end of this presentation, is apoptosis is the neatest way for cell to die, cleanest way, okay? Cell dies, no issues with body. One cell is gone, we'll put another one there. Okay, there is no inflammation, there is nothing else. So that is the characteristic of P53. So let's talk about the growth advantage. So what's going to happen with tumor suppressor gene mutations? Genetic mutations will actually provide growth advantage to these cells. So this will cause this. This will allow selective growth of the cells inside the tissue. The mutations of these genes, specifically tumor suppressor genes, will make cells grow better, grow faster, because they're abnormal. They divide much faster. So that will give them an advantage inside the tissue. They will actually grow more and faster than normal cells. So let's look at this. What actually mutations are associated with growth advantage and development of cancer? Proto-oncogene mutation, remember, will result in the activation of oncogenes. Does it make sense? Proto-oncogenes gets mutated, and now it becomes oncogene, gets activated, and causes cancer. Now, this is a dominant mutation. That's the bad news. So which means you need just one copy of the gene to be messed up, that the whole proto-oncogene will turn into oncogene and cause issues, lead to malignancy. While tumor suppressor genes are inactivated by mutation. So they're not activated by proto-oncogenes. Tumor suppressor gene get mutated, and what do? They inactivate. They do not work properly, okay? And this is actually recessive mutation. You see how our body is set up. Is tumor suppressor genes are really important things in your body. So the mutations are usually recessive mutations. And this means that you're going to have both copies of tumor suppressor genes mutated in order to stop the tumor suppressor gene. This is a fail-safe mechanism. It actually ensures that your tumor suppressor genes are protected. So it has to be mutation of both copies of the gene. While proto-oncogene, to be active, you just need one single dominant mutation, and it becomes an oncogene. Now let's look at this. So cancer process is a development of cancer, as you know, is a stepwise process. It's not something that occurs overnight. So several alterations must occur before malignant transformation of the cell. So what does that mean? So what happens is if you have single mutation, it doesn't mean you will automatically develop cancer. You have to have additional mutation. So what does that mean? You have DNA mutation. Okay? Let's start it from there. Gene mutation. But your proto-oncogene is working. No proto-oncogene, sorry. Your tumor suppressor protein is working. It's going to figure out something is wrong. It's going to stop your cells, kill them. But what's going to happen? You have a mutation, and then you have a mutation on your tumor suppressor gene. Now, the bad cell goes through the whole process. So that requires several mutations to develop cancer, not just one. Now, interestingly, childhood cancers require fewer mutations to become malignant, and inherited mutations will predispose patients to cancer. It will not cause cancer in most of the cases, okay? It will predispose patients to cancer. So let's talk about the growth regulation. We know that the survival and development of normal cell is controlled by a complex set of biochemical signals, and cancer cells are free from this control. So what does that mean? Is your cells are constantly controlled by growth factors, okay? And they're controlling their growth, they're controlling how they move, they're listening to the laws, okay? They obey the laws. What's going to happen? Cancer cells don't care. They do not react to growth stimulation. They will react if it's going to help them. If it's not going to help them, they will not respond, okay? So that is the characteristic for it. So the cancer genesis is that the mutations of key genes have to accumulate over time. So what does that mean? Is you're going to have a mutation of one gene, then another gene, then the other gene, and then as it accumulates, it will lead to cancer. Now, these cancers, cells will produce a progeny of cells that will not react to growth regulation. So they will not react to any kind of regulation. But accumulation of mutations, guys, will actually take years to develop. It's not going to immediately happen. Okay? And then you might have exogenous and endogenous processes, something from the outside. You know, you have higher incidence chance of, for example, developing a breast cancer. But then you go on the, let's say, you go on the hormone replacement therapy. Now you're adding problems to already genetic mutation that will occur, probably cause another genetic mutation, which will now cause probably development of cancer. So it takes years to develop. Now we know if DNA is not repaired through these mutations, this will result in the development of cancer. So these are different theories that tell you what happens and how cancer, how cancer evolves. We don't know exactly, but there are different theories that tell you this. There are two major theories I'm going to talk about. This is known as clonal evolution model. So what does that mean? You have a single cell, okay? And this cell gets mutated. And it goes through a cycle. And this cell, what's going to do now? This cell will now reproduce. There's going to be more cells. But this mutation gives a growth advantage to this cell. It will grow better than the rest of the cells. So now suddenly the cells of your tissue will be overtaken by the mutated cells. Okay? The cells that have growth advantage. But still not cancer. Okay? Now, what's going to happen? Now the next mutation occurs and affects already mutated cells. And it gives those cells a growth advantage. So now you have second mutation, and you have new cells that will have growth advantage. So they will now grow and push all the others. So after several steps of mutations, you will have cancer. Okay? You have one mutation, gives cells growth advantage. Now these cells take majority of the tissue. Now you have new mutation, giving now new set of cells a growth advantage, now growing, and as these mutations progress, eventually all the cells become cancerous in the tissue. So this is a clonal evolution model. So what are the hallmarks of cancer? What will tell you that certain tissue or certain cell is actually cancerous? Is this. Is number one, it's going to become self-sufficient. It will not depend on growth signals. It's just going to grow. It will become insensitive to growth inhibitor signals. If the body tells her, stop growing, it's going to continue growing. It will evade apoptosis. They're going to get signals, kill yourself, and the cell will not do it. It's going to stick around. Next, it will acquire a limitless replicative potential. It will replicate. You know, she will avoid good old Hayflick's limit. Say, forget it, I'm doing it. The next thing is, it will have to sustain angiogenesis. Now I'm explaining this a little bit more to you. Why? As the cancer cells grow rapidly, what do they need to grow? They need oxygen. They need blood. Okay? They need blood supply. So it will actually have to stimulate the development of new blood vessels and a blood supply to the tumorous cells. And this is known as angiogenesis. So they will need to produce enough blood vessels to feed cancer cells. And let me explain you what happens. In a normal tissue, you know, you will have blood vessels all over. You will have blood vessel to a single cell if you need to. But no, normally, in normal cells will produce angiogenetic inhibitors. They will produce compounds that will inhibit formation of new blood vessels. You don't need it. You need too many blood vessels. So if there is a need for more blood vessels, cells will now release angiogenesis activators. Angiogenesis means formation of blood vessels. So they will now activate formation of the blood vessels. So interestingly, tumor cells will produce two very potent angiogenesis activators that will activate formation of new blood vessels to the tumor. And these are known as vascular endothelial growth factor, VEGF, and basic fibroblast growth factor, or BFGF. These two guys will help cancer get blood supply to it. They will push the body to produce the more blood vessels towards the cancer. Good example, and this is kind of interesting example, is the P53 mutation will prevent normal production of the molecule known as thrombospondin. And thrombospondin is angiogenesis inhibitor. It inhibits the formation of new blood vessels. So a mutation of P53, they will result in decreased production of thrombospondin and more blood vessels produced towards the cancer. Now let's talk about stem cell theory. This is the second of the two important theories that tell us how the cancer might develop. We might think that some cancers or tumors actually contain stem cells with indefinite replicative potential. You know, stem cells are crazy. They can divide, no problem. They can stop dividing, be hiding somewhere, and they can start dividing again. So that is the characteristic of stem cells. Okay? They're very similar to adult stem cells, these cancer stem cells. And these cancer stem cells are self-renewing. And what cancer stem cell can do? It can produce a tumor, but it can also produce a blood vessel for a tumor. Okay? Because cancer stem cell can make any other cell if it wants. Okay? So the presence of cancer stem cells is another theory that tells you the development of cancer. Interesting for stem cells is they're kind of resistant to drugs, very much resistant to drugs, and they express specific markers that are characteristic for stem cells on their surface. Why do we use this theory of the stem cell? Simply because you remember, some people will be cancer-free for years. You can't find a single problem in them. And then suddenly the cancer reoccurs. Why? Because we think that cancer stem cell was somewhere, stem cells group, a population, that was waiting to be reactivated by environmental factors, okay? And that will result in cancer. So let's talk about the tumor progression. Now we know that one problem in cancer cells is that they actually have tendency to metastasize. They can disconnect from the attachments, travel to blood and go somewhere else. That's the problem. What is the problem? They will obtain genes that allow them to break tissue structure and invade basal membrane. So what does that mean? Is they can first detach from from the rest of the cells. You remember those extracellular matrix we talked about, gap junctions, all that stuff we mentioned? They can actually form proteins that will disconnect them from it. They will just dislodge from the tissue. Now they will travel to distant location, and what they're going to do is now they're going to invade the tissue. They will go through the basal membrane of endothelial cells, so the blood vessels, and they will go into the tissue. This will enable cancer cells to migrate between different tissues in the body. And now, of course, whenever they form a tumor, that they will require a large amount of blood vessels. So tumors will accumulate in a mass, will need to induce the growth of the blood vessels and activate angiogenesis. So this growth of the blood vessels ensure that the tumor gets enough oxygen. Now, this is an important slide. Why? Because I want you to learn this table. Why? Because it's very, very important. It's important because it's going to pop out on boards. It's going to pop out through your next 2 years all the time. Now, what is it? These are the inherited gene mutations that are associated with different types of cancer. So what does that mean when an individual is BRCA1 mutation positive? Okay? It has mutation of BRCA1. It makes sense. It tells you that this individual has several-fold increased risk to develop cancer. But you're going to tell your patient doesn't mean you will get a cancer. Okay? But you will have much higher incidence of developing cancer. Interestingly, whenever patients have these mutations, please understand is that mutation is inherited through germline, through actually original spermatozoid and the ovum. So it actually is carried in every single cell of the body. So you can find it everywhere. There is a mutation there, okay? And again, the presence of mutation will do this. It will increase the possibility of developing subsequent mutations on top of that mutation, leading to cancer, okay? So, and again, what is the problem? Let's talk about mutations in tumor suppressor genes. You have an individual who has, some mutation on some tumor suppressor gene. I'm not talking big guys. I'm talking smaller guys that I didn't mention. I'm not talking about P53 and others, but smaller guys. So does that mean that they will immediately develop cancer? No. They will have to develop additional mutation, kaboom, to develop cancer. However, if there is a mutation in proto-oncogenes that is carried through germline, is usually, and I'll tell you sadly, it's usually associated, you will not see those patients. Why? Because it's not compatible with life most of the time. So not all of the time, but most of the time. So actually, the fetus will die in utero before birth. That's the way how we prevent an abnormal, that's how the abnormal progeny is prevented in that sense, because if the proto-oncogene is activated. Oncogene, you remember, oncogene doesn't let things form. Okay? It causes cancerous cells, abnormal division. When is the highest division? During embryological development. So these will be abnormal fetuses that will be actually, they will die in utero. So let's talk about it. Let's talk about the environmental factors, because there are tons of environmental factors associated with cancer. Now we have different types of environmental chemicals that can cause cancer. They can be called genotoxic, which means they are toxic to your genome. They usually interact with DNA, cause mutations in different critical genes, or they can be non-genotoxic, which means they can lead to cancer, but not through interaction with DNA. Through some indirect interaction, not direct interaction, that will eventually lead to cancer. So it's a very multi-step process of these environmental factors affecting it. So let me just explain to you a really scary thing. Chemicals that we know who have no cancerogenic potential. Okay, so what does that mean? Your hand lotion is not cancerogenic. You use it. Well, put it on your hand. What's going to happen if you choose to sleep in it? Sleep in it for the next 10 years. Okay? There's a chance that that non-cancerogenic compound might, from prolonged exposure and all this stuff, abnormal exposure, will actually lead to cancer. Okay? So that is the characteristic of it. So chemicals with no cancerogenic potential need significant amount of time in order to mediate tumor progression. So everything that you have has some kind of potential to give you mutations. But you have to, for example, choose to eat laptops. I don't know, or some weird stuff, you know. So, but that is it. But if you are overly exposed, that can cause to cancer. The next thing is this, is benzopyrene adducts. We're going to mention them. They're actually found in cigarettes, directly bind to DNA and cause mutation of P53 in your lungs, so cause cancer, okay? The next one are aflatoxin and hepatitis B. Big board question, Dr. D is not going to ask you, but aflatoxin is found in certain types of fungi that actually live in grain siluses, okay? So they actually can be toxic, and they can cause liver cancer if people do not check their grain for aflatoxin and make bread with it, okay? So there are places in the world that ended up with huge liver cancer epidemics because of aflatoxin that was found inside the grain, okay? So also hepatitis B is a virus that will activate and actually cause a mutation of P53 leading to liver cancer. UV light is bad. So, you know, you spend a lot of time on the beach under sunlight, or UV light, even worse, in those tanning booths, it can lead to a mutation of P53 leading to skin cancer. HPV, we already talked about it. Human papilloma virus will cause issues with P53 and lead to cancer. Now, another thing that can cause cancer is mutation of the enzymes in your body that remove toxic cancerous substances. So, for example, Dr. D has a fantastic group of enzymes. And, you know, Bob has less enzymes. The enzymes in his liver are not that good at toxic body. You're going to hear from these enzymes much more in depth in block 2. So what's going to happen is, I eat cancerous stuff, but my enzymes are better. They work much better, so I have a less chance of developing cancer. Bob does not. His enzymes don't work well. So this is a characteristic. So there is a mutation that affects the enzymes, detoxifying enzymes, for normally cancerogenic substances can lead to cancer. So for example, cytochrome P450, that's a huge group of enzymes. Have you talked about it already? No? You will in biochemistry. These are the enzymes that detoxify your body galore. They're a huge group of enzymes. They're called cytochrome P450. And they are the ones who will help you eliminate junk you put in your body every day, or junk that's made in your body. So if P53 have mutation, have better chance of developing cancer. You might have accumulation of glutathione transferase. You know, guys, you heard a lot of favorite things. This is one of my favorite enzymes. Glutathione transferase is another enzyme that is involved in detoxification, and N-acetyltransferase. So we see that different things, environmental factors can cause cancer. Let's talk about the types of cell death. You know, my research is kind of weird. Why? Because I developed an interesting, I would call morbid interest in cellular death, okay? Why? Because I love to, you know, first in, you know, all of you who did research, I was very successful in destroying my cell colonies. Very successful. For a lot of reasons. For example, it took me 3 months to figure out that my sleeve is touching my cells and all the weird stuff. Okay, again, I'm a doctor, so it took me time to become a PhD, trust me. So what it is, you know, you kind of go through this. So I kind of said, oh my God, cell death, that would be cool to do. So that was a research. My research was in apoptosis. But let's talk about cell death. There are 2 ways the cell will die in a body. Kind of cool, guys. It's like CSI, Dr. D style, okay? So, number 1, you know, necrosis. And I want to have those glasses. What's the name of that actor, guys? Remind me, with the cool glasses that does the, used to do that. Caruso, yeah. I'm going to do Caruso on you, okay? So, the types of cell death, you know, ta-da, there has to be music. But, so, necrosis, what is it? It's a passive process. It's a stupid way to die, okay, guys? So what is it? But it is the most common way to die, okay? So it's induced by cell injuries. Cells just get injured, boom, you know, explode and die. Okay, kind of cool. So what is it? Their membranes will be ruptured. They will spill their guts all over the, you know, all over the tissue. This will now induce inflammation. Inflammation will pick up all the junk that's all over there. There's a cleaning crew. Okay, fine, they die. That's the normal necrosis. Apoptosis is a smooth way to die. Okay, why? Because this is the way to give exit to these cancer cells. You die, nobody knows. You know, it's like a mafia style. You don't hear anything, everything is fine. You know, Bob is here no more. I have nothing against Bob's. John is here no more. Okay, but so this is it. So apoptosis is a normal physiological process. It's involved in death of individual cells. Single cell dies, not groups, not 15, 20. One dies. So if one cell dies and nobody is there to hear it, did it die? I'm just joking. It just came up to me. So, but it's true, you know. So it is this. It's actually a Zen way to die, okay? So, and it's very important. There is no spillage of cellular content. Cells don't spill their guts. They just shrink, okay? And there's no disturbance of this process will lead to cancer, because that's how we kill our cancer cells. We don't want a huge mess. We just tell them, you know, you're not a celebrity anymore, come here. So it's done, you know, gone. So that's what it is. So they will have something called bleb membranes. The membrane develop blebs. That's a key word. If I ask you a question, give you a question, cell has bleb membranes, man, I know what it is. It's like apoptosis, you know, it didn't burst. There is no inflammation. No inflammation. So let's talk about necrosis. It's a passive pathological process induced by injury or disease. It's passively cell die due to necrosis. Why? Because the whole group of cells will die. How do they die? Among others, the death can be associated, for example, in ischemia, lack of oxygen. Cells will decrease the amount of ATP produced in them. So because of the injury or something else. What's going to happen when ATP goes down? There is a very important pump I mentioned on the first or second lecture called sodium-potassium pump. Sodium-potassium pump does not work because it needs ATP. And you remember what is sodium-potassium pump important? In maintaining the cell volume. So the cell will become fatter and fatter with water and boom, burst. Okay? So that is the characteristic of it. Now they will spill their content, activate inflammation, and the process is completed within several days. I'm going to show you one clinical example. It's kind of cool. Normally, cytoplasm is full of an enzyme known as lactate dehydrogenase. Have you heard that in biochemistry yet? You will. They'll mention this. It actually is involved in conversion of glucose to ATP. So cells have a lot of LDH, lactose dehydrogenase in it. And you will find some in blood, but very little. Why? Because it's in cells. So what's going to happen when you have massive injury? Cells die, huge group of cells die. They will release that lactate dehydrogenase in the blood. So you suddenly start seeing spikes of lactate dehydrogenase. It tells you, oh my God, there is some serious necrosis somewhere happening. There are groups of cells dying somewhere, let's look at it. So now, apoptosis is actually an intracellular suicide program. It happens step by step by step by step, okay? So we're going to talk about it. Cell will receive the signals that support the survival and ensure that they live on. Think about it. Cells have to listen constantly to soothing music, okay? Why? Because for them not to commit suicide. So what does that mean? They have to be constantly reminded, you should live. You live, you know, be happy, live, you know. So they have constant signals that are constantly telling them, what's going to happen when these signals stop? No music. Boom, I kill myself. So that is actually how cells work. It's weird, but true. So when these signals stop, they will undergo apoptosis. So membrane is actually, during apoptosis, important that membrane is preserved. They do not burst. The membrane is preserved. You remember this. You remember the membrane phospholipid known as phosphatidylserine will go from the inner leaflet to the outer leaflet to signal that cell is ready to die. Okay? Now membrane portions will bud off from the membrane and will form these blebs. Okay? Blebs of membrane. Now, cells will lose symmetry. They will kind of start looking funkier. They will look like almost like dried out, and they will detach from the tissue, okay? Now, their mitochondria, inside them will lose a really nasty molecule that is important in oxidative phosphorylation, known as cytochrome C. Now, cytochrome C will be released into the cytoplasm. Now, cytochrome C will remain in the membrane blebs and will help process of apoptosis. Now, apoptotic cell chromatin will actually condense. It's going to, you remember, normally chromatin is all dissolved. Now it's going to condense in corners. Now, phagocytic cells will go around and find the cells they want to die that will have phosphatidylserine on their surface. They're going to attach on them, and boom, they're going to eat it. They're going to eat it and burp. Done. Okay? Cell died. Okay? So that is it. Now, they want to say, is there any mess? Your phagocytic cells have eaten away the cell that wanted to die. And what do? In order to ensure that there is no inflammation, that your skin, that your area doesn't get inflamed, that no more immune cells come, These phagocytes will release interleukin-10 and TGF-beta, transforming growth factor beta. And these guys inhibit inflammation. So they will cover up the tracks, these macrophages. No problem. Very smooth way to die. Now, so let's talk about the elimination of damaged cells. So this is it, guys. Situation 1. You have a cell that is damaged beyond repair and infected by virus or starved. So what's going to happen? Tumor suppressor P53 will stop that cell during the cycle. You're going to say, this is something wrong with this cell, okay? And will induce apoptosis. Now, how does it do that? Now we're going into explanation how P53 induces apoptosis. We're going to go through steps, and I'm going to explain it. So protein P53 will bind to a gene promoter of a specific pro-apoptotic protein that causes apoptosis, known as BAX. And this will now trigger apoptosis. Result, you get rid of the cell. It dies quietly, no drama, gone. Okay? Now, if there is a mutation of P53, oh my God, this cell that has mutation will pass through the P53 and will now start growing. There is no apoptosis. It goes to cancer. So, occasionally I kind of lose these things. I don't know why. Or maybe I have to be in certain positions. So that's why I'm here, guys. I'm just joking. So let's see. Okay, no. I'm going to click on it. Not a biggie. So, now, let's go next. So this is next. This is why apoptosis is important. Oh, now it goes. So, apoptosis is important because it gives you those cute little fingers that you have, okay, and toes, okay. What's important? It's very important. It's a normal process. Actually, it's not only connected with drama. Apoptosis is actually very selective during embryogenesis, which will get rid of the skin between your fingers. Will get rid of that skin that will help you open your eyes, and develop palpebrae, which you can look through it. So it's very important because it removes excessive cells during organogenesis and limb development, and it sculpts the newly formed body. So that's very, very important. So what's going to happen if apoptosis did not work during this period? You're going to become very popular in a frat house. Why? Because you're going to have extra nipples, for example. You'll be like, look, guys, I have an extra nipple. Oh my God, this guy is going to be leader of our group. Okay? Ladies, that's how guys think most of the time. Okay? Doesn't matter what his grade is. He has extra nipples, man. So that's it, So that's the idea. But actually, it's kind of okay. I would have patients say, I have extra nipples, I want to remove them. I was like, why? Geez, you're going to be like cool. You can tell that to your kids, spread it around, it's healthy. It's not a big deal. But what it is, so if there is a failure of localized apoptosis during development, so if there is a problem with apoptosis, of course, people can be born with these web fingers. They have to go under surgery and stuff. So that's why we see that apoptosis is quite important. Now, apoptosis is important because it maintains homeostasis. We mentioned this. We have to have same number of cells die and new numbers of cells will be replicated. So that is important. So there is a constant balance between cell division and cell death. So billions of cells will die every day and they need to be replaced by new cells. So what's going to happen if there is disturbance of this equilibrium we mentioned? We go to cancer. Okay. So now, there is a gene that is kind of cool, and I put it here because it shows you how molecular biologists, how many of you worked in molecular biology labs? I know. You probably know how excited it is. Definitely not. So, so this is actually a sonic hedgehog gene. Truly named. It's actually a sonic hedgehog gene. What does that tell you? There was some sad, depressed molecular biology PhD student finding a new gene and calling it, I'm going to call it sonic hedgehog. So it is. It actually is a sonic hedgehog gene. So it's very important. And, and I want to meet the guy who did it or gal who did it. It's just super awesome idea. It'd be like Dr. D's awesomest and best gene. Something like that. You give them like a cool name, Or, like, I don't know, like my cat Oscar gene. So, and that's going to be a gene for cuteness. I'm sorry. but that's it. So SHA gene is sonic pathway. Guys, one thing, I'm going back. I'm going to run out of time. But, I'm going back. Like, I'm just looking at my cat. I don't know how many of you got cats. But dogs love you. Dogs do stuff for you. Cats don't do a thing, But you love them, and you get so excited about the fact they just look cute. And I said, can you imagine if our lives were like that? Just stand there like, we love you. Why? Look at your cute little ears. We'll feed you. You know, this would be awesome living like a cat, man. It's like, you know, it's like, meh, clean my litter box. Okay, no problem, sir. Yeah, I'm hungry. Give me food. I don't want to eat this stuff. You know how you know the cat person from dog person in a pet store? This is it. You know, dog persons go buy food for dog, put it, and leave. Cat person's like, I don't know if it's going to eat this. My cat doesn't eat chicken. He eats chicken. But he likes salmon, specifically wild caught. But, you know, it's just this like, you know. And I had a dog and a cat, so I know now. I'm a cat person, but I love dogs too. You know, dogs will eat anything. You just give them, they'll eat it, you know. But the cat is like, no, I don't think this is okay. You know, it's like, we're going down on the quality of food. You know, it's like, yeah, you've probably been there. You know, you put stuff like, here you are. It's like, kitty. Like, bah, I don't eat it. Yeah. And, you know, for some reason, you know, you could, like, leave them for 3 days. They'll eat it eventually. But you can't, because they drive you nuts at night. They're like, nah, 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 nah. It's like, you know, this is awesome. So sonic Hedgehog genes are actually send anti-apoptotic signals to the cells. Those are that music that's been sent to the cells not to die. When they stop, when there's a mutation of them, what's going to happen is that, so they send anti-apoptotic signals to the cells. So failure to receive the signal will lead to apoptosis, okay? So what's going to happen? If you have sonic Hedgehog genes expressed in some tissue, it will make cells survive much longer and longer and longer. They will not die. They will be constantly told to survive. And that can lead to malignancy. So let's look at the apoptosome. So we're going to talk about a little bit the process of apoptosis. So irreparable damage is sustained to cellular components or DNA. And this will initiate internal cell death program. So what is it? You remember that the BAX will be activated. We mentioned this BAX protein. BAX will be inserted into mitochondrial membrane and will enable the exit of cytochrome C out from the mitochondria. And cytochrome C will now activate formation of apoptosome, which is the early structure that indicates the beginning of apoptosis. Now, how does that happen? Cytochrome C will activate APF1 adapter protein. I know you're kind of getting really tired with all these abbreviations, but that's what it is. Cytochrome C will activate APF1 adapter protein, which will now turn on enzyme inside the cell known as caspase-9. Caspase-9 is a mean bugger, okay? So caspase-9, when it gets activated, will actually, so cytochrome C is released by the insertion of BAX into the mitochondrial membrane. It will now activate APF, which will in turn activate caspase-9. And caspase-9 will now cleave and destroy cellular proteins and DNA and will progress cell into the cell death. Result is death by apoptosis. Now, this is really cool, guys. There are actually receptors that can tell cells to go and die. There is a self-destruct button on a cell. You can activate them, cells die. So these are known as death receptors. And they're actually part of the tumor necrosis factor receptors. They can initiate apoptosis from external signals. External signals can initiate apoptosis. So individual members of this family will be recognized specific ligands. Please understand that not all tumor necrosis factor receptors will cause apoptosis. But the ones that cause apoptosis have inside of the cell this known as death domain. Kind of cool, okay? So they will have inside the cell this death domain that will now initiate the process of apoptosis. When they get activated, death domain lights up. When death domain lights up, cell dies. So what is the characteristic of these death domains? They will actually interact with enzymes known as caspase-8 or 10. We mentioned caspase-8 as a very mean player. So the caspase-8 will be activated and eventually lead to cell death. So let's talk about how this happens just briefly. So you have a fast death receptor. You remember the death receptors that's there. Member of TNFR superfamily will engage the apoptotic cell death, will induce apoptotic cell death when actually it engages with a Fas ligand, Fas ligand. Fas ligand will bind to Fas receptor. Boom, activate death domain, cell dies. Why is this important? What's going to happen if you have some really highly infected cell floating around, full of viruses? Okay, it's virus replicated in it. Do you know, if you guys watch the Minions movie, you know those like green minions that go blah, blah, blah? Yeah, that's what it is. It's a weird cell. It's going all over, okay? So that strange cell that is going all over, that's full, needs to be destroyed. How are you going to destroy it? You are a T cytotoxic lymphocyte. So going around and see this funky looking minion going around. You want to kill it. You don't want it to go around. You don't know what's going to happen. So what does it do? It has fast ligand. So this T cytotoxic cell will come in, and its ligand, it will attach to the fast receptor of this cell. It's going to press that self-destruction button. When it does it, the cell will go into apoptosis and die. Okay? And that will prevent, for example, spread of viruses or everything else. So let's explain this again, just a little bit more in depth, is that we have to understand that how do apoptotic cells occur? We have to understand in all of our cells, there is a constant struggle between the empire and rebels, okay? There's a constant trouble between the pro-apoptotic proteins and anti-apoptotic proteins. And they have to be maintained in balance, okay? So if they're maintained in balance, or if you want cells to survive longer, you can do anti-apoptotic proteins that will actually prevent apoptosis. So this balance is very important. So what are these pro and anti-apoptotic proteins? They're members of BCL2 family. Actually worked on these. So BCL, specifically BCL2 family. So you have two sets of proteins. You have pro-survival members, which are known as BCL2 and BCLXL. And you have pro-death members that will stimulate the death, and they're back and backs. So let's look at this. So if there is a death receptor signaling, will result in activation of caspase-8. We mentioned, remember that nasty caspase-8 that gets activated. Caspase-8 will cause cleavage of BID to TBID, which is the adapter protein. And TBID will now help backs to translocate from cytoplasm into the membrane of the mitochondria. Okay? It will kind of lodge itself into the membrane of mitochondria, backs and back. And they will now cause mitochondrial membrane to become permeabilized. It will kind of open channels on the membrane. And membrane will now release cytochrome C and really cool protein known as SMAC Diablo. Okay? Is it good to have a lot of SMAC Diablo? No, probably not. So that's it. So from mitochondria, cytochrome C and SMAC Diablo are released. And now cytochrome C will cause all the problems, all the issues. It will start cell into the formation of apopsosome and death. And SMAC Diablo is going to do something. It's going to inhibit. It's going to inhibit pro-survival members. So SMAC Diablo is going to go there with a knife and kill good old BSL2 and BSLXL. Okay? It's going to say, you're not going to stop this. We're killing it. Okay? And the cell will die. Okay, guys, that's pretty much it. So good luck. I'll see you in next block. Kind of done with me now. Yeah, I'm going to miss you too, but don't worry. You know, I'll see you soon. So, you know.